So I'd like to say hello to Camellia and hello to Lydia. Is everybody else here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Caitlin's on her way, isn't she? She's doing questions. 13. Who am I missing?
Ellie.
Oh, Ellie. Anybody seen Ellie?
She's in.
Yeah, right. Look, okay. Mention on one of your assessments, it was mentioned that there's practical work. There's one practical we've done that I didn't mention yesterday, never thought of. The hemosatometer. So don't forget that, okay? That's practical work, so make sure you have a look over that. We will, whenever we come back after Easter, we'll spend time preparing, going over things, okay? Hi. So, now, so Ellie, what I was saying is, don't forget to look over the hemosatometer. We were talking about practicals yesterday and it never entered my head, so that's something that, is practical we did this year. So, respiration. Does anybody want to fire any things that they want me to go through first, or will I just start? Do you want me to sort of recap on things? Yeah. Yes? Yeah. Okay, right. So, we always tend to talk about aerobic respiration first of all, so that's what we'll do, okay? So we'll start off by talking about glucose then. So what do you know about fructose and glucose? They're both sugars. Many carbons do they have?
6.
And they're isomers, they both are C6H12O6. Okay, now you don't need to write all this down. But I'm just basically going to go through the flow diagram and so on. So feel free, you can write things down, or you can just annotate your notes, or you can just sit there and take it all in, whichever you find works. And I'm recording this, okay, so if everybody's happy, so that Camellia can see it back in Hong Kong, and if you want to, then you can go back over it as well. So, they're both isomers, so they're both C6H12O6. The way I remember this first stage is it's fructose bisphosphate. What does bisphosphate mean? It contains 2 phosphates. So the way I remember is it's taking the phosphates from ATP. And therefore we're making ADP. So remember adenosine triphosphate. So there's your 3 phosphates. You're taking one of those off and you're having 2 molecules. So the 2 phosphates are joining with the glucose to make fructose bisphosphate. Okay? So that's the first stage. That splits into, whoops. So we had a C6H12O6 and 2 phosphates. Now, I don't know exactly what the formula is, but basically that's what it is. You've got the same carbons, hydrogen, oxygen, just added in 2 phosphates. That splits in half, so you get a 3-carbon compound, and each one of them has 1 of the phosphates. So that's all that's happening there. Okay, then NAD picks up some NAD, well, picks up some hydrogens, becomes reduced. Some inorganic phosphate is needed to make the bisphosphate part there. And the same thing is happening over on the right-hand side there. Glycerate phosphate is still a 3-carbon compound. And the final stage then is where this is converted into pyruvate, which you know is also a 3-carbon compound. We've got 2 molecules of ADP picking up phosphate to make 2 molecules of ATP, and that's an energy-rich molecule. Same thing is happening over here. So again, NAD is picking up the hydrogens. Okay, so what do we call that whole thing? What is that whole process from glucose down to pyruvate? Glycolysis. And where does it happen? Yeah. Okay, so this is glycolysis. And it happens in the cytoplasm. Hi. So if we look and see, we're starting with glucose, and we end up with 2 molecules of pyruvate. Okay. Well, we need 2 molecules of ATP, and we need NAD. We need 2 molecules of NAD. Okay. We're also needing some ADP as well there. Now the NAD is important because we come to that later whenever we look at anaerobic respiration and we say that we need to regenerate NAD. And that's because we haven't mentioned oxygen there. Glycolysis doesn't need oxygen to, anywhere in any of those equations. Okay, so it happens in aerobic conditions and also anaerobic conditions. But for it to keep on going, there's only so many NAD molecules in a cell. So say there were 100 molecules in a cell. So if you have lots of glycolysis happening around the cell, whenever your 100 NAD molecules become NADH, glycolysis will stop. Okay, so there needs to be some mechanism to regenerate the NAD, and that's what the anaerobic respiration making lactate or making ethanol does. Okay, it regenerates the NAD to allow glycolysis to happen in anaerobic conditions. So what are we making? In terms of what are we getting? We're making 2 molecules of NADH and we're making 4 molecules of ATP. But I've already said that we're using 2 ATP. So really, so if we go from pyruvate here, then we've got the link reaction followed by the Krebs cycle. So the link reaction is basically changing pyruvate into acetyl coenzyme A. Okay. Acetyl coenzyme A is a 2-carbon molecule. So we lose a carbon here in the form of carbon dioxide. So coenzyme A needs to be added there to make acetyl coenzyme A. We lose a carbon dioxide molecule. We also need DNA to pick up the hydrogens there. Okay? So in the link reaction, we've got pyruvate getting converted into acetyl coenzyme A. We are producing carbon dioxide, NADH. Now if we were to go... Okay, the Krebs cycle. In the matrix of the mitochondrion, this is where this happens, you've got a 4-carbon acid there. It picks up the acetyl coenzyme A to make a 6-carbon acid. This cycle used to be called the citric acid cycle. That gets converted into a 5-carbon acid, which then gets converted into a 4-carbon acid. Each time, if you lose a carbon, you lose it in the form of carbon dioxide. Okay. Now, the coenzyme A comes out there and it's basically just recycled from there. It can be used over and over again. So we've got 6-carbon acid. In terms of what happens here, really, you need to know that there are 3 NADs becoming 3 NADH2s or 3 NADHs. It doesn't matter what you call it there. Okay, there's 1 FAD becoming FADH2. Okay. And there's also 1 ADP picking up phosphate and becoming ATP. So in the Krebs cycle, we've got 3 NADHs or H2s, we've got an FADH2 and we've got an ATP. And we've also got 2 molecules of carbon dioxide. But remember that you've got 2 link reactions and therefore you're going to have 2 Krebs cycles for every molecule of glucose. We said, so I've highlighted there's NAD picking up hydrogens becoming NADH. If that doesn't happen, glycolysis will stop at this point here. So if we run out of NAD here and here, glycolysis would stop at that point there. Right? So what do we need? Let me see here. Our net gain, what is our overall gain of ATP? Well, we said we made 4, we used 2, so our net gain is 2. NADH, how many molecules did we make? 2. Okay. Over here. Okay, with no ATP, but we've got 1 molecule of NAD, but remember, we've got 2 pyruvate molecules, so we're going to make 2 molecules of NADH. Okay. The other thing there is that we get a molecule of carbon dioxide for each link reaction. So we said for each Krebs cycle you get one molecule of ATP, but remember, if we're talking about what happens when one C6H12O6, one glucose molecule, gets broken down, one per cycle, we've 2 cycles, so we've got 2 there. We've 3 NADHs per cycle, so multiply that by 2 and you get 6. And the other thing we've got is FADH2, and we've got 1 per cycle, so that's 2. Now, if we look at the amount of ATP, we're only getting 4 molecules of ATP made. And we call that at substrate level phosphorylation. Phosphorylation is adding phosphate group. Okay, so that quite often that's called substrate level phosphorylation. Okay, we've got 10 of these and we've got 2 of these. So this is, we know, where do those molecules go? Electron transport chain. For every 3 NADHs, NADH that carries hydrogen to the electron transport chain, how many molecules of ATP do you get? 3. So that's going to give us 30 ATP. For every FADH2, how many ATP molecules? 2. So that's going to give us 4. So from the electron transport chain, we're getting 34 molecules of ATP. We say that is by oxidative phosphorylation. Where does the oxygen come in the electron transport chain? At the very end, yeah. So if we need oxygen there to accept the hydrogens to make water at the very end, so hence the name oxidative phosphorylation, because you've got ADP, you're adding a phosphate on, hence the term phosphorylation. So altogether we have 4 at substrate level phosphorylation, so 2 in glycolysis, 2 in the Krebs cycles, and then our other 34 comes from the electron transport chain. So from 1 molecule of glucose, you should get 38 molecules of ATP as a result of aerobic respiration. That's what you should get. 36 might be closer because it actually takes some energy to move molecules in and out of the mitochondrion. But as far as you're concerned, 38 is the answer, and that you've done all the sums and it works out. Makes sense? Okay? Do you want me to go through the electron transport chain? Yes, okay, I see a few nods here. Glycolysis. So the electron transport chain is essentially a series of carrier molecules. So we've got okay, so these are carrier molecules, electron carriers or hydrogen carriers. They are at progressively lower energy levels. So anytime you talk about the molecules, you should say they're at progressively lower energy levels. All right? So NAD NAD carries the hydrogen to the electron transport chain. That's how it gets there. When it passes the hydrogen on, the flavoprotein becomes reduced. All right? But the NAD becomes oxidized. So sometimes, you'll see these complicated, flavoprotein picks it up and becomes reduced. Okay? I'm not going to bother with writing all that stuff there. Okay? But basically, it passes hydrogen on to the flavoprotein. At that stage, enough energy is liberated to allow ADP and inorganic phosphate to become ATP. Okay, so because there's a drop in energy levels, that energy is enough to cause ADP to become ATP. Flavoprotein then passes the hydrogen on, but at this stage, it goes to the coenzyme Q molecule. It becomes reduced. Flavoprotein is no longer reduced. It becomes oxidized. So we can talk about redox reactions. When it picks up the hydrogens, it becomes reduced. When it passes it on, gets rid of it, it's oxidized. So coenzyme Q picks up the hydrogens. At this stage then, the hydrogens separate or they dissociate. So a hydrogen atom has got a proton in the nucleus, and then there's an electron floating around there in the orbit. That's what a hydrogen atom looks like, a positive and a negative. They basically split. So the electron separates, and it's the electron then that gets passed down here. Gets passed down these stages here. The hydrogen is what you're left behind. So if you lose the electron, you're left with the positive proton in the middle, which is basically the hydrogen ion. So the hydrogen basically is floating about in the matrix of the mitochondria. Meanwhile, the electrons are getting passed down here. And so you're left with the electron getting passed down from carrier to carrier at some stages. So at this stage here, we get another molecule of ATP from here, and we get another molecule of ATP from here. So we've got 1, 2, 3 molecules of ATP. Right at this bit here, the hydrogens and electrons come back together again to make hydrogen. And the final acceptor is oxygen. So if we've got oxygen there, oxygen accepts the hydrogen and becomes water. Okay, so oxygen is the final electron acceptor. If you don't have oxygen, the whole thing will stop. Because the first time, if you've no oxygen, the first set of electrons will get down as far as cytochrome oxidase, and then they'll be stuck. So that will be reduced. The next electrons will get down to the cytochromes, but they can't be passed on to the cytochrome oxidase because it's already reduced. The next ones will get down here, and the whole thing stops. So we're saying normally this will happen in aerobic conditions. Every molecule of reduced NAD or NADH that enters the electron transport chain will yield 3 molecules of ATP through oxidative phosphorylation. The hydrogen is passed down a series of carrier molecules that are at, what's the phrase? Yeah, progressively lower energy levels. At 3 stages, enough energy is liberated to make ADP plus P give you ATP. You can say that the hydrogens are passed down the first couple of carrier molecules. Now, you can name them if you want. But then after coenzyme Q, the hydrogen dissociates. And it's the electrons then are passed down the carrier molecules. Oxygen is the final acceptor. And it accepts the hydrogen to form water. Now, during the Krebs cycle, we saw FAD feature. It picked up hydrogens, became reduced. It enters at a different stage. It passes its hydrogens onto the coenzyme Q. So it's missing out that first stage. And so therefore, you don't get this molecule of ATP. For every FADH2 that carries the hydrogens, you get 2 molecules of ATP. Okay? Any questions about all of that? Back to if there's no oxygen, this whole electron transport chain will stop. Okay, so we mentioned anaerobic respiration. So we said that glycolysis doesn't need oxygen. So we're starting off with glucose. You get 2 molecules of pyruvate. How many ATP do you get? What's the net gain of ATP? 2. So you only get 2 molecules of ATP made. How many do you get with aerobic respiration? 38. So anaerobic respiration only gives you a tiny amount of energy compared to aerobic respiration. But it still allows energy to be released. So there are some organisms that, where, for example, yeast, you know that yeast respires anaerobically, it makes alcohol, and you're aware of making wine and brewing and making bread and stuff like that. So anaerobic respiration, you're very well aware of that. Yeast will actually respire, it's what we call a facultative anaerobe. It will respire, if there's oxygen there, it'll respire aerobically. Once the oxygen runs out, it'll respire anaerobically. There are very few organisms that only respire anaerobically. There's only a handful of bacteria that will respire anaerobically all of the time. Your muscle cells can respire anaerobically, but it's only short periods of time, and it's only if you're sprinting, okay, just for, a number of seconds, a minute or two, and not be longer than that. What's the whole point of it? It only gives you 2 molecules of ATP, but that's on top of the normal aerobic respiration that's going on in your muscle cells. Okay, so your muscle cells will be getting oxygen from your blood and your breathing and so on. So your muscle cells will be getting a supply of oxygen, so there will be normal aerobic respiration occurring. But the reason why some of your muscle cells will respire anaerobically is because they're not getting enough oxygen. So this is extra respiration that wouldn't normally be happening. So it's an advantage, it's extra, it's over and above the normal ATP that's produced during aerobic respiration. All right, so we've got 2 molecules of ATP. We know that NAD picks up hydrogens, becomes NADH. How many do we have all? Is it 2 or is it 4? I think it's 2. Okay. So going to happen is somehow we need to be able to recycle the NAD. Normally, we've just said what happens. Normally, the NADH carries the hydrogens to the electron transport chain, it passes the hydrogens on to the flavoprotein, which passes it on to the coenzyme Q, which passes it on to whatever and whatever and whatever. And so normally the NADH passes the hydrogens on and it becomes NAD. It goes back, is used for glycolysis to keep glycolysis happening. That's not possible if there's no oxygen, because we know. So how do we basically get rid of the hydrogens from our NADH? So there's two ways. If it's yeast cells or plant roots, pyruvate basically reacts with the NADH. Pyruvate takes the hydrogen, so the pyruvate is reduced and it makes ethanol and carbon dioxide. So you'd be aware, you would have learnt the word equation. Glucose gives you ethanol, carbon dioxide, and small amount of energy. You would have learnt that GCSE. What have we got here? We're starting with glucose. We're ending up with ethanol, carbon dioxide, and a little bit of ATP, 2 molecules of ATP. The NADH, which came from here, this is the NADH that we're talking about. It reacts with the pyruvate. Basically, the hydrogen reacts with the pyruvate to make ethanol and carbon dioxide. Okay? So that happens in yeast cells and in plants. So in terms of plants, if a plant got waterlogged, so if there was a lot of rain and the soil all got waterlogged, there was nowhere for the rain to drain away to, all the air spaces in the soil get filled with water. There's no air, there's no oxygen. The root cells will start to respire anaerobically and they make ethanol. And they can do that for a number of days, but if it was to stay waterlogged for a long period of time, that the level of alcohol will build up and will start to damage the cells. Okay. What happens in your muscle cells? So the NADH reacts with the pyruvate. The hydrogen reduces the pyruvate to make lactate. Okay. And obviously you've regenerated the NAD and that allows glycolysis to keep going. So hence in muscle cells it's glucose, gives you lactate, and there's no carbon dioxide there. Small amount of energy. So basically we're saying it's the NADH. The hydrogen is just used to reduce the pyruvate. In animal muscle cells, it'll make lactate. In plant cells and in yeast cells, it'll make ethanol and carbon dioxide. The important thing is the NAD is regenerated, and that will allow glycolysis to keep on going, to continue in those cells that are respiring. Lactate and alcohol, they need to be broken down because, as we said, if they build up, they'll damage the cells. And so after you've stopped sprinting, you'll keep on taking extra breaths, deeper breaths, getting more oxygen there, and that's to break down the lactate that's in your cells. We talk about repaying the oxygen debt there. Now, one other thing. So just to summarize, glucose, pyruvate, acetyl coenzyme A, and then we've got the Krebs cycle. And then obviously we've got the electron transport chain. I'll just put it at the side. 6 carbons. Pyruvate, how many carbons? Acetyl coenzyme A? 2. This will make a 6 carbon, 5 carbon, and then a 4 carbon here. So, carbohydrates. So your polysaccharides are made of lots of monosaccharides. And basically, most things will get broken down. So if you're taking starch or some kind of complex carbohydrate, it generally gets broken down into 6 carbon units. So whether that's glucose, fructose, galactose, whatever, they are all sort of C6H12O6. So the carbohydrates will go down this route here. They will enter at this stage here. In terms of proteins, well, proteins are made up of amino acids. So I suppose if we hydrolyze, so hydrolysis, breaking down, that happens during digestion, you get amino acids. What do you remember about amino acids? Well, there are 20 different amino acids. They have got, let me see, the amino group at one end, the carboxylic acid group at the other end, and then the R is what we call the variable group. So that could be like CH3OH, it could be anything, all right? So you could have this being broken down into, basically the protein could be broken down into amino acids, and some of them might have 3 carbons. So if they have 3 carbons, they will enter around the pyruvate stage. If they've got 2 carbon units, they'll enter here. If they've got 4 carbon units or 5 carbon units, they'll enter at different stages of the Krebs cycle. Okay. In terms of lipids, when you eat a fat or a lipid, it gets hydrolyzed, in other words, digested. What does it get broken down into? Sorry? Fatty acids and glycerol. So glycerol, we'll look at it first. It's the easy one. It's 3 carbons. And then hydrogen's coming off there. 3 carbons. So if it's got 3 carbons, it can join at this stage here, just above pyruvate. Fatty acids, you might have 16 carbons with lots of hydrogens and so on. There is nowhere where we've got 16 or 14 or whatever. So what happens to the fatty acids? Anybody remember? Well, basically they're chopped into 2 carbon units. So there's 2 carbons, 2 carbons, 2 carbons, and so on. And so they enter, where there are 2 carbons, they enter here. So if you had a lipid, it's going to have 3 fatty acids. And each of those fatty acids might be broken into 7 units or 8 units. In other words, it's going to go around the Krebs cycle 7 times or 8 times. And you've got the glycerol, which is going to go in at the pyruvate stage. That gives you an indication of just how much more energy you get from a lipid molecule than you would from protein or from carbohydrate. Okay? So you need to know where these they're called respiratory substrates. They are the substrate, the starting, like the ingredient for respiration. All right. Respiratory quotients was something, I'm not sure if we talked about it or if we just brushed over. If we look at this, which part of respiration can occur without oxygen? There's only one stage. Glycolysis. So if we look at glycolysis, this is glycolysis from glucose down to pyruvate. Only carbohydrates go through glycolysis. The proteins, the earliest they join is at the pyruvate stage. Glycerol joins just at the pyruvate stage. Fatty acids join further down. So if there's anaerobic respiration occurring, it can only be carbohydrates. For it, RQ, respiratory quotient, volume of carbon dioxide produced divided by volume of oxygen consumed. Now, if we look at the word equation for glucose as a respiratory substrate, C6H12O6. The balanced chemical equation. Volume of carbon dioxide produced, well, we've got 6. And oxygen consumed is also 6. So the RQ value, if they give you that equation, the RQ value is 6 divided by 6, which is 0. I've got a sheet here, which I'll give you. Number 3. Where it's on the other side. Okay, on the sheet that I've just given you then, it's got the fat tripalmitin. This came up in an exam question a while back, and it said we've got a fat, gave the name of it, and then gave the equation. And the question said, calculate the RQ value. So RQ value is carbon dioxide over oxygen. So carbon dioxide, oxygen is going to be 102 divided by 145. So when you get your calculator out, what do you get? What do you get?
0.703.
Yep, 0.703, which should, yeah, 0.7. The RQ value for carbohydrate is always 1. RQ value for fat or lipid is always 0.7. What is it for protein? For protein, it is 0.9. And those are 3 figures that you have to learn and know. Some organisms might be converting, so organisms that are going into hibernation, that are converting carbohydrate into fat for storage, that conversion process actually gives off carbon dioxide, and so therefore it's going to put the top up. So if there's something else going on, like the conversion of carbohydrate to fat for storage, it's going to put that up. That's going to cause the RQ value goes slightly over 1. Another thing that causes it to go over 1 is if there is anaerobic respiration occurring. So, see at the bottom there, I've got a summary table, respiratory substrate. What do we say it is for carbohydrate? What's the RQ value? 1. So you write 1.0. And what I want you to do, so we've got respiratory substrate, RQ 1.0, I want you to write conditions, stick another column, conditions, and you're going to write aerobic. Fat is 0.7, and again, that's aerobic. Protein, 0.9. This next one, you can just put in brackets, conversion to fat. So we're talking about organisms going into hibernation. And then it can go anything from 2 up to 7. The conditions are anaerobic, so we're going to stick that over here. Okay, so everybody got anaerobic written down there. So in, that table there, the substrate is carbohydrates. Okay, so you've got a couple of wee questions on the right-hand side of that page. Oh, sorry, we've been underneath there. I mean, you're going to get things like organisms where the RQ value works out at 0.95 or something like that. And that's indicating that there's a mixture of respiratory substrates. Okay, so there's definitely carbohydrate there to get it over and above the, the 0.7 or the 0.9. So you'd say there's a mixture of respiratory substrates in aerobic conditions. So there might be, you would say, carbohydrate and protein or carbohydrate and fat. You don't know which it is because you've only got like an average figure. So let's look at the right-hand side. It says pea and castor oil seeds contain different types of food store. Looking at germinating seeds. I tell you what, we've only really got 2 minutes left. You can have a look at that and you can have a look at the question on the other side and try to do that for, I don't know whether this is in your booklet or not, but have a look at that for tomorrow and we'll start off tomorrow by going through that. Caitlin, you missed at the beginning, I don't know if I said to you, when I talked about practical work, I forgot to remind you about the hemocytometer yesterday, so make sure you don't forget about hemocytometer when you're revising. And then tomorrow we're going to look at photosynthesis. Again, with photosynthesis, are there any aspects or do you just want me to go through, do what I did today? Yeah, okay. At the beginning then.